Hi guys. So we in class today talked about the five-finger model for how we know when evolution is occurring in a population. So that's for the pinky small population. Then we're going to have evolution probably happening if there is non-random mating, so our pointer or our ring finger. Middle mutation, so we're going to have genetic mutations producing new alleles, pointing in the right direction with our index finger. That's going to be immigration happening. So if we're having transfer, gene flow of new alleles, or if we have some sort of advantage or disadvantage to the organisms, that's our thumbs up or thumbs down. So natural selection is happening. If any one of these is happening, then we are going to be having evolution happening in that population. Okay, so what if a population is not evolving? So in that case, that means the frequency of alleles and genotypes in a population in that gene pool are going to remain constant unless they're acted upon by another agent other than sexual recombination. And we can describe that in using something called the Hardy-Weinberg equilibrium. Okay, so the Hardy-Weinberg equilibrium is a way of describing that equilibrium that's happening when evolution is not occurring. Okay, so we need to make sure we have the conditions met for something to fall into this Hardy-Weinberg equilibrium application that we can use. And so that's no selection. These are all related kind of pretty much the opposite of our five finger. So no selection, no mutation, no migration, a large population, and random mating. So this is going to ensure that we're able to follow this Hardy-Weinberg equilibrium. If any of these are happening, that means we are going to have at least microevolution occurring at some extent, and therefore we cannot use it doesn't meet the assumption, so we cannot use the Hardy-Weinberg equilibrium. Okay, so what is the Hardy-Weinberg equilibrium if things are met? It's two equations that will be given to you on the test that are describing the alleles in a population. So first of all, this is used in examples where there's only two alleles for a trait. So in this case, we have color, we have purple and pink, but those are the only two alleles. We have purple is dominant to pink in this case. If there's something else where there's a third allele, we're not going to be able to use that in this example. But our first equation is that the frequency of all of our dominant alleles plus the frequency of all of our recessive alleles are going to equal 1. So we can think about this as like 100%. So if we add up all the individuals with, or all of the alleles that are dominant, all the alleles that are recessive, that will be 100% of the alleles in the population. Okay, so this is a pretty straightforward equation right here. The second equation that we have is this, and that is looking at each of the individuals. So if we have a homozygous individual, so that means, and they're dominant, there's going to have two P's. So they have to have one P and another P from their other parent in order to be homozygous dominant. So we represent that by P squared, and that's the probability of having both P's as genes from the two parents. Then we have the possibility of having a heterozygous, so that's having a P and a Q. So we're going to have P times Q, but we also need to multiply this 2 times. If you can think about if we have a Punnett square that is homozygous crossed with a homozygous. So we have homozygous dominant crossed with homozygous recessive. We're going to have half of the time occurring, or 2 of the 4 chances where we can have one that is heterozygous. So that's where we're getting this 2PQ from. And then the last option is our Q, so our recessive phenotype, that we get our Q squared because it's the possibility of having a Q and then another Q recessive genotype. Okay, so all of this added together, which is our homozygous dominant plus our heterozygous and our homozygous recessive, should also equal 100% of our population. So we can use these two equations to figure out the number of individuals that are going to have each specific type of genotype. Okay, so here's an example. Suppose a plant population, red flowers, where R is dominant to white. In a population of 500 individuals, 25% show the recessive phenotype. How many individuals would you expect to be homozygous dominant and heterozygous for this trait? So because we are given the recessive phenotype, because we're able to observe that, we can't observe which one is homozygous dominant versus heterozygous. We can just observe a dominant phenotype. So we have to use this Hardy-Weinberg equilibrium equation to figure out the number of individuals. So how this works is we do know our white flowers are recessive, and that means they're going to be lowercase r. And we know that 25% of the population is having that genotype. So we can then conclude that Q squared, which remember is our recessive genotype, is going to account for 25% of the time, so 0.25. So our first step is to take this and we're going to figure out what just one Q is. So we're going to square both, or do the square root to both sides to figure out that Q equals 0.5, because we went from Q squared to just Q. So now that we have Q, we can figure out what P is by using this first equation. P plus Q equals 1. So if Q is 0.5, we can then figure out that P is also 0.5. In this case, both of them are the same, but in other cases, it will be a little more complicated. Our next step is to now put these into the correct part of our equation. So for a homozygous dominant individual, that's P squared. We're going to put 0.5 squared, so 0.25 or 25%. And then we can figure out the number of homozygous dominant individuals by multiplying that percentage by the total population, which in this case is 500. So we can figure out that there are 125 individuals that are homozygous dominant. We can do the same thing for our heterozygous, where we have 2 times P times Q. So we have 2 times 0.5 times 0.5, and if we do that, we will get 0.5. So that means 50% of our population is heterozygous. So 50% of 500 is 250 individuals. So we can use this information to figure out the number of individuals that have each genotype. If, again, making sure that this is a population that is following the Hardy-Weinberg equilibrium assumptions, which is basically that it's just not evolving. So the big thing is figure out what the recessive is and then have that equal to Q squared. Do the square root to figure out what just Q is, figure out what P is, and then you can plug it in to figure out what your percentages are for each genotype. Okay, that is how we do these problems. So you can go ahead and start working on those practice problems. I hope this is helpful. Bye, guys.